For this last lesson on how proteins are made, you are going to need those 5 colors we used before: yellow, orange, red, green, and blue. And you're also going to need purple and pink for this one. The process we're going to look at here is called transcription. Now don't get this confused with the word translation. It's very easy to confuse these two words. Transcription has the word script in it. So when you see script, that means writing. Think copying. Think of a scribe. You know, back in the Middle Ages, the scribes would sit there and they would write copies of things by hand. They would make copies. So if you kind of think of this as a scribe writing copies of something, actually there's a cartoon that sort of uses that, and the scribe, the writing would be the messenger RNA, and we're going to meet the scribe that actually does the writing. So this is transcription. Well, let's start out talking about where it takes place. And we're going to draw a simple cell right here. And let me tell you, this is going to be simple. Easiest drawing yet. Right here, we're going to draw a cell. Just like that. It can be kind of oblong to fit into space. Make sure we leave space over here for something else. And we'll label it cell. And in this cell, we're going to put the biggest, most visible organelle, and of course you know this is the nucleus. And in the nucleus, we're going to put squiggly DNA. And I'm sure you knew this as well. DNA. And I want to mention something about the nucleus here. DNA is a very important substance, as I'm sure you know. It contains all the information that a cell will ever need, and stuff that it will never need. So the DNA has to be kept safe. If you lose your DNA, you lose your cell. So the plasma membrane around the nucleus is double thick for double safety. So we're going to draw a little circle around a little piece of it, and we're going to enlarge it. We'll go like this, showing that. And then I'm going to write something right here. So we want to come over on this side, and let's show you a close-up of this membrane. What I want you to do real lightly is draw as if you're going to do that phospholipid bilayer, which that's exactly what it's going to be, but you're going to make 2 of them like that. Now we can shortcut. Remember, you can sit there and do this whole thing, or if you want to kind of do that curly Q thing I showed you, and try to shortcut it, that's fine. We know what it's supposed to be. I'm going to use the curly Q method here. And then I'm going to shortcut these tails and just make some lines across like that. But it's obvious what it's supposed to be. So the nucleus has a double layer. So, right, the plasma membrane looks like this only. I mean, it has a double layer of these phospholipid heads. So actually the nucleus, if you're just talking about the phospholipids, it's got 1, 2, 3, 4 layers of them. And there's actually a little more space in between here. These are kind of crowded together a little bit too much. But the main thing is just to know that there's 2 of them, and that's really important. And there's little holes in them. I'm not going to put one here, but we'll get to that later. There's little holes in the outside here where things can go in and out. And you imagine the pores are pretty elaborate. They kind of look like these like basketball hoop things. Inside the nucleus, there's also a dark patch. I don't know, this might be something you're less familiar with. So just draw something like that. And I want to show you, I got an actual photograph of a real cell here showing this. So this is a nucleus. We're really zoomed in here. This isn't the cell. You might think this is the cell and that's the nucleus, but this is actually the nucleus. And so all the DNA would be around in here, and it doesn't look like double helices, does it? And this dark spot is called the nucleolus. And this one, you can see the whole cell, and I believe this is the nucleus here, and then this little dot would be the nucleolus. So what does the nucleolus do? Well, it's made of DNA, mostly. It's not really a separate thing. It's just an area of the nucleus that's a bit darker. The DNA is more dense there, a lot more of it packed together, and there would be RNA and some proteins floating around too. So let's label this nucleolus. And this is where ribosomes are made. And if this is the only fact you remember about the nucleolus, that's okay. So let's talk a little bit more about DNA. Let's go over here, since this is so very important. Let's make sure we give it its due. We're going to compare it to RNA. We saw the structure of RNA in the last drawing, and of course DNA is very similar, as you are aware. So we're mainly going to look at the differences. And of course, again, here's a little picture of it, and we're all very familiar with the picture. One thing I'd like to point out, in our drawing we're not going to be able to do this, but you see how it's kind of not just the same. We're just going to make all the wiggles the same, but you see how this gap is a little bit deeper than this one? This is like goes in shallow, and this is deep, and that's shallow. Well, they have names for these, the major and minor groove, but just want to point out that a unit of DNA isn't one of these. A unit goes from here to here. This is a full twist, like that. A figure 8 is the full twist. Okay, so what are the differences between DNA and RNA? Well, we'll do the obvious one first. DNA is a double helix. With 2 strands. It's like a ladder with 2 sides. And of course, as we saw in the last drawing, RNA is a single helix. And it still is a helix, so a lot of times in drawings it looks like a line. It technically still is a helix with 1 strand. Now you can have double-stranded RNA, okay, it's weird, it's like in viruses and stuff, but generally RNA will be a single strand. Okay, so something I think we already touched on before, but let's officially write it here. DNA has thymine. I'm going to make this a capital, kind of underline it to make sure we get the T there. Thymine, because when we write it, we'll just put T. DNA has thymine. RNA has uracil, U. And remember that they're very similar. Thymine is basically uracil with a CH3 stuck on it. Okay, number 3, DNA has, as its sugar, deoxyribose. You may remember this. Oops, I got to back out a little bit. Where we looked at the ribose sugar, right? This is plain ribose, and the difference is just here. See, this is OH and H, so when you deoxy it, you get deoxyribose. So that's basically the only difference between the two. So RNA has ribose. And then I want to add DNA is found only in the nucleus, whereas RNA is found in all parts of the cell. So let's see how messenger RNA is made. And this guy is going to be our little protein gadget called RNA polymerase. Right there, RNA polymerase. Now this is a protein gadget. And part of the way I know this is a protein gadget is I look at the word and it ends in ASE. So I know that it is basically an enzyme, a protein enzyme. It's going to be in the center here. We need a roughed out circle. Leave some space here. Leave a little space because we want to write a few words. We want to label at the bottom. And then leave a little bit of space, maybe on the edge here, at least say 1, 2, 3, 4, 5, 6 or 7 centimeters. Like underneath the word where that word DNA stops, right before here. You know, that would be a good guideline. Okay? So just kind of to give us a guide of where we're going to be headed, but it's not going to be this simple because this is a little gadget. Okay, I think you can see that. Okay, first thing is pretend you're going to draw in the middle, but we're not going to draw in the middle. We're going to draw a little triangle, but we want to put it slightly off center. In fact, I'm looking at my sketch here. Make this, give us a little bit more space. Make it a little bit oval, just slightly. Don't make it a perfect circle. I'm just looking at what all we have to fit in here. You may need to kind of go out a little bit. That's why I always say keep your lines really light at first, because then that gives you the option of changing. So you may want to shoot for more like an oval here to get everything in. Okay, so instead of drawing it right in the center, just move off a little bit from center like that and make a triangle like this. And we might want to alter this and make it more pointy towards the end, but right now that's good. This is going to be like the scissor thing. When something comes through, it's going to hit that and split off like this. It's going to be the splitter. So we need a tube to let the thing come in like this. Imagine that this is the entrance here where the thing's going to come in and get split. And then over the top like this. Now the real thing is a little more bumpy than this. And actually I'm going to show you a picture here in just a second. Ours is going to be nice and smooth, so it's easy to draw. Let me show you what it really looks like. Okay, this is a computer-generated image, but it shows you this is going to be DNA, this is going to be messenger RNA, and there's the gadget we're drawing. There's another picture of it. Okay, and so this is, see here, this says Mg, magnesium atom, right at the center. One of the working parts at the center involves a magnesium ion. And here is the protein structure. Now, when we looked at the ribosome, remember I said it's kind of looks like it might be made of protein, but it isn't. But here we actually see there's the helices. You know, these curly Q helices. So this is made out of protein. And it's got the empty space in the center here. This is where the stuff's going to feed through. Our picture's going to look more like this. We're going to kind of have it be kind of on its side like this. Our DNA is going to feed in straight, but we're going to do like a cutaway. Like so here you can see what's going on inside, and like in this picture here you can't see what's going on inside. Right, so we're going to have to do kind of a cutaway so that we can see what's going on right here. Okay, so that will be good enough. And then, and here, so this is going to be our feed-in tube, and then down here we'll have, this is another solid section, our cutaway view, something like this. And then there's going to be another inlet coming in here, but we kind of, this is like a wide area down here. It's going to be more like a funnel shape. Wide and then narrowing. Just only go to about there. And then we're going to need an exit over here. So go like that. And then we can have a more narrow exit there. And then we need an exit right here. So we need to put DNA feeding into here. It's going to get split, and then it's going to get rejoined here. So this is how we're going to do it. Since this is a splitter, you can imagine the two strands like this. Put them right in the middle there. And it doesn't have to be tight in there. You have to do that. Okay, so it looks like it's going to get split. And then outside here, though, where it's not in yet, this is going to be regular DNA. And we're going to be really lazy. We're not going to draw it with that major minor groove. This is good enough. If you just go up and down a few times, that's fine. There we go. That's way good enough. Okay, like that. But make sure you get a couple of these because we're going to like put the correct little things on there and label it. And then it's going to go through like that. And then this strand is going to come down here. Now leave some space because we're going to have, let's see, messenger RNA being formed. So make this come straight across. Make sure we give this enough space here. That's why I draw in pencil, so I can adjust. Okay, so we got this, it's going to be here. And I do want to get this sort of matched up. Let's see, we'll put the strands, if we make the messenger RNA be pretty tight down to here, that'll be okay. We'll keep this up. We do need enough space in here. We're going to make the little nucleotides showing between here. And then it's going to go run along there and up and out. And then we're going to do the same thing over here. We're just going to go up and down and up and down. This one's going up like that. There. And if you want to make your splitter look like it's going right like that, you can. We're not going to do anything else. Or you could write splitter or scissor. This is what I mean by scissor. But of course you can see it doesn't really, it's not like it snips it this way. It just splits it. And I want you to use purple. You're going to have to follow these exact colors for this one. I'll give you some leeway on these, but let's use purple because we're not using it for any of the nucleotides. And let's make the backbone. Remember the sugar phosphate backbone. We're not going to show all the sugars and phosphates. We don't need to have all that in this drawing. So let's make the backbone purple. And then when you get to here, make the top one, let's leave this for a second. When it goes out, make this one purple. I'm going to mix some pink, and it'll make sense when we get to the bottom of the page. We'll be explaining why. But in here, in these two sections, some of the DNA is going to be filler and need to be cut out. Now, it's important. It has a job while it's in the DNA, but when we make messenger RNA, there's going to be little sections that have to be snipped out. So let's just pretend we've got some of those. We're going to make the extra things that we clip out, we're going to make pink. So we have a good section, then we have this filler section. Like I said, it's not junk DNA, it's not waste or anything. It's important. There's a reason it's in there. But when you copy it to messenger RNA, the intent is that these will be removed. Okay, it's like when you get something in the mail, something fragile, and it comes with, bubble wrap and things wrapped around it, or it comes with a package insert that's molded to be around that object. Well, you can't say, well, that's why'd they even put it in the box? That's not part of my product. Well, if they hadn't put it in the box, your product would have gotten destroyed by the time it got to you, right? It was very important to have that bubble wrap there while it was being mailed. But then once you get it, you don't need the bubble wrap. It's kind of a little bit like that, although these things aren't exactly like bubble wrap. But at any rate, they're important. They have a job, but then they get edited out. So down here, let's do the same thing. Now, we're not going to try to match up. Like, we know our drawing isn't going to be like scientifically PhD level perfect, right? Okay, just the fact that you know that there's these little sections in here, that's pretty good. So let's see, we'll make well, I'll be approximate on pink and purple, make them look pretty much the same on the bottom here, but if it's not identical, that's okay. So remember, this is just the backbone here, sugar phosphate backbone. Okay, now what about the rungs? This is where you're going to use, on the DNA, we're going to use the green and blue for C and G, right? And I'm going to use the yellow again for A. In fact, I think I'm going to switch to a yellow that's a little more tan, it's a little easier to see instead of my bright yellow, but still identifiably yellow in comparison to other colors. And we're going to use red for thymine. Remember, we're going to use orange for uracil, but we're going to use red here for thymine because this is going to be DNA. So this is the part where you have some leeway. I don't care what order these are in, just as long as you use those four colors, and we always match green and blue, cytosine and guanine, and we always match yellow and red, A and T, adenine and thymine. Now, the number of rungs we put in here is, well, fairly important. Not to be totally exact, but this is one twist, right? And it will have about 10 rungs. So you might not see, if you imagine this is 3D, one of the rungs might be sort of hidden in the twist here. So you don't have to go 1, 2, 3, 4, 5, 1, 2, 3, 4, 5, right? One of them might be in here and here. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, something like that. So about that many. And you can put them in whatever order, just as long as your blues and greens match. And to speed it up a little bit, I'm going to put like all my green in first. Green, 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 like that. And then I'm going to put the blue in matching on all of them. It's a little more efficient when you do it this way, I think. Let's see, blue, green, blue. It didn't start that way. And then I'll put in where I want the yellow. See, you can just see that a little better on camera. I hope it still looks yellowy. Maybe I can go over it with the other yellow. Just afraid this yellow is not showing up quite well enough. Red, red, red. That's just peeking out there a little bit behind. Then when we get to here, these are going to split. So this thing is going to come down here, right? And it's going to still have these rungs on it. So it doesn't matter what exact order here, still whatever red. And they don't have to match up perfectly with these pink and purple. Like I said, this is not like a PhD level drawing. This is just meant to show some basic things. Let's see, I'm going to put a few greens on here. So if they're not spaced perfectly or something, don't worry about it. On the top also, this one is going to loop up over here. And I'm just going to, well, I guess if it's pointing that direction, I guess we can put the, I think it's going out that way. And while we're putting in these colors, let's think about what's going on. When messenger RNA comes in to make a copy, which strand here is it really copying? Let's say you want to make a copy of this strand, and messenger RNA comes in and starts matching. For blue, it puts green, and for yellow, it's going to put orange, and it starts matching. What you're going to get down here is not a copy of this. You're going to get the reverse. When you start matching down here for the messenger RNA, you're going to get the opposite. For every green, you're going to get a blue, and for every red, you're going to get an orange, or for every yellow, you're going to get an orange. So, oddly enough, the strand that I'm working on now, this top strand, this is the one that is being copied. This is the information the cell needs. It's encoded on this top strand. So, since this was matching, right, this is the opposite of this. These were matched up, and if there was a green down here, there was a blue up here. Now they're pulled apart. Now the green's down here, the blue's up here. But when we start putting together messenger RNA, the green will be matched up with the blue. So if there's a blue up here, there will be blue on messenger RNA. So this is going to be a copy of this because it's a reverse of this. Right? This is a reverse of this, and this is a reverse of this. Therefore, this is a copy of that. Okay, enough said. Hope you get the point here. Going back out, just put some more so make a whole bunch of these bases, and you'll see then we're going to put on a, oops, I almost used the red, orange, those will be yours, so kind of like that. And that's going to represent our nucleotide. It's yellow. So what's a nucleotide? Well, the colored part, right, is the base of adenine, uracil, guanine, cytosine, and the purple represents a sugar, ribose, and a phosphate, PO4. Alright, so they're not all going to be lined up perfectly. This is a bit too orderly for real life, but you get the point. There's some blues. Now, this is where the critical action happens. There's something here, a complicated thing. When I say complicated, I do mean these things are complicated. I can just show you. I mean, this is just a very overly simplified drawing of this protein and all these things that are all hooked together. Okay, so it's really complicated. So there's a little thing here that allows the nucleotides to match up and then hook together to form an RNA. So for example, we'll just take this one here. Say this is the first one. I see blue. So a green one is going to stick right here. And then the next one I have looks like green. So maybe I'll stick it with a blue. And then here, see, this one is a yellow, so it's on its way. Let's say it's not stuck yet. Here it is coming in, ready, or yellow would match with orange. The orange one is coming in to match with this yellow right here. Okay, it's going to, and then there's a green one, so there'd be a blue one lined up here. So this just must be a sea of these nucleotides. Amazing, they all find their way over. So once they get close like this, you see how they're going to join. That one looks like a blue. So the next one, there's yellow. So we're going to need orange, not red. I keep trying to use the wrong one. So then that's going to hook up. You see how we're starting to form a new backbone there? So this backbone is going to be the messenger RNA backbone. And then, let's see, I'm going to stop worrying too much about being super accurate here in just a minute. As we get on to here, I'll be like, okay, orange, orange, that one hooked up, there. Okay, so there we go. It's going out with its various bases. Okay, so this would be our messenger RNA, right? mRNA coming out. I can kind of shore that up a little bit. Now we're going to add a note right here. When this comes out, it's actually not quite ready to be used. And in fact, I should have added some in here. We got these pink places. Okay, because it's a copy. And it's called pre-messenger RNA. When it comes out, it is called pre-mRNA and must be, I'll say edited, E-D-I-T-E-D. To edit something is to make little changes to it. It has to be kind of tweaked, trimmed a little bit. So it must be edited. Now down here, let's put the title, just to remind ourselves, RNA polymerase. And ACE means ASE means it is a protein gadget. And technically / an enzyme. That's the correct word. Okay, so just noting that that word actually tells you something about what it is. Okay, let's right here, nucleotides, waiting. And our DNA, I think we can remember this is DNA, but let's remind ourselves of our code up here. Let's write adenine, thymine, cytosine, guanine, and then a little bar of color for each one. So if someone else came and looked at our drawing, they would know what the colors mean. And then down here we could also put the orange for uracil, for RNA. And let's write a note to ourselves here that this is like a loop. If we put a line down here and a line down here, and then an arrow, there are about 10 rungs per twist. We already said that, but I thought it'd just be nice to label it. And then the last thing that's important to label is the ends. Remember, I told you that they need to keep track of which end is which. It's very important. Which is the front end, which is the rear end. That's very important. Well, the front end here coming out, the leading end, is called 5 prime. I think we saw that before too. 5 with a little apostrophe mark. That's the 5 prime end. So this would be the 3 prime forming. In DNA, they keep track of the strands because one of them runs 5 to 3 and the other goes 3 to 5. So let's make this one, let's see, let's follow it. I'm looking at my model. Okay, so let's make this one running on the bottom here. This way, and then follow it out. There, this would be the end right here. So that would be the 3. So the other one would be 5. And then follow, let's follow the 3 prime end. So the other end of this strand, I'm going to keep track of this one. Up, down, up. It's the one that comes down. And then it goes up here, right? So this would be, see, that was 3 on that end, so it would be 5 on this end. And so the other one would be the 3. So it's a big deal to the people that work with DNA. Now let's see what we're going to do with those pink and purple sections. Let's go down here. I'll zoom in. And we'll deal with these edits that have to be done. So we're going to draw this brand new piece of pre-messenger RNA. It's still got the pink and purple sections in it. And I want you to draw it like a loop. This is going to be the front. We're going to loop around and go like this because we don't have enough space to go all the way across because we have something we're going to put over in this block. So we're going to have to bend it a little bit like this. We're going to do like a hairpin turn. So we're going to have a special thing right here. In fact, we could just even go ahead and really, really lightly draw in a circle. We're going to put a special cap on the front end of it. So we'll leave room. That will be where the cap goes. So then this will be the actual pre-messenger RNA, and then I'm going to make it without the nucleotides showing. Remember, I said you can decide how you want to draw RNA and similar things according to what you want to show. You can change and draw it a different way. And that's what we're going to do here. We're not going to draw little sticks sticking out for the nucleotides. I'm going to write them just as letters. So this is going to be my first section, and if you remember from, I think we did it in the activity for sure, what's the first thing on the messenger RNA that's going to mean start? A, U, G. There's your little start codon. You know what's going to be on the front. Isn't that cool? You actually know part of the RNA code now. So then we don't know these. We're just going to fill in with whatever. C, C, G, A. Yours doesn't have to be similar. Just remember use U instead of T. A, U, U, A, C, G, C. And then I'm going to turn so that my letters are all spacing up. A, U, C, G. And then we can use a stop codon at the end if you want to. And I'm going to use UGA for stop. UGA. Remember, it's getting read this way. UGA. And that means my little stop codon. And then some of these sections are going to be purple and pink. Now, the purple ones, they stay on. We don't want to trim off the start and stop. This is going to be very important. We're going to keep these. And then just kind of randomly choose a couple. Let's see, there's going to be a keeper. And then we'll have a pink section. Now the pink sections are going to represent something called an intron. And maybe I'll make mine only one codon long. Of course, this is ridiculously short. You know that in real life it would be much longer than this, of course. But this will be long enough to get our point across. Okay, so there's going to be another good section. Let's say 2 more codons worth. This is going to be a keeper. The keeper sections are called exons, and the throwaway sections are called introns. And this is one of the worst naming scenarios I can think of in biology. To me, this is awful. Why in the world did you call the good things you want to keep in the exons and the things you're clipping out the introns? Because obviously the things that stay in, there's things that want to stay in, you keep them in, and then the things that you want to get rid of should be the exons because they're exiting, right? It's so obvious. You should have called the things that stay in the introns. No, they had to have it reversed. And this, for the life of me, I can't remember this. I hope you do better than me. I'm trying to think of a mnemonic. How in the world am I not going to get it backwards? The best I could do was intron, intruder. You don't want the intruder, something like that. Very difficult. So the pink sections here, what we're going to clip out, these are called the introns. I almost said the wrong thing. The introns, the intruders. Intron is intruders. And then these purple ones that we're keeping are going to be the exons. Okay, I'm going to make another pink intron here. Purple section. And yours do not have to line up with mine. Just so you have some pinks and some purples, that's good enough. In fact, it's not going to match what I had above. It's not going to come close. Okay. And then the rest of it's going to be purple. Okay, so this is the way that it came out of that RNA polymerase. And then as soon as it gets out, there's some other little worker robots standing by. You can't see them in the picture. They'd be like sitting here. There's other little robot guys. They're just waiting. You don't have to add them, but just so you know, there's these little robot guys waiting to add some things to it. It needs to have a special cap on the front called a 5' cap. And this is what it will look like. Well, actually, I'll show you. How do you put a cap on something at the molecular level? Well, this is what you do. Here comes the RNA. You've got sugar, phosphate, right? Phosphate, sugar, base, I don't know, adenine, cytosine, whatever. Phosphate, sugar, base. And this has been going on for a long time, right? This is the very end of the chain here. So it's phosphate, sugar, phosphate, sugar, phosphate. So this is where you would get another sugar, phosphate, but this is it. This was the end. That's it. So they add 2 phosphates, another sugar, a guanine, and then this thing called a methyl group, CH3. We're going to see this again, a methyl tag on the end of that. So that's how you cap off the end. So this is just kind of fun. You don't have to memorize this, of course not. But it's fun to just go circle, circle, circle. And then put an upside down, I think it's upside down is how it goes, pentagon for the ribose. And then we're using rectangles for bases, G for guanine. And then you have this methyl CH3. Now, I do want you to know that this is called a methyl tag. methyl. Now, why is it called a methyl tag? Well, hopefully this will jog your memory. How do you count carbons? Remember way back in lesson 2? It's amazing how that stuff in those first couple lessons, it keeps coming up. Meth. Meth is how you say 1 when you count carbons. Meth, eth, prop, but, pent. There's a special way to count carbons. And meth means 1, and there's the 1, C. So this thing, CH3, has a special name. It's called the methyl group, and here it's called the methyl tag. And we're going to see this again. This is a way you zip DNA shut. If you want to keep it closed, you're going to use these little things. These can act like little paper clips, little binder clips to close things up. So we'll see those again. And so this is G. If you want to write guanine, you can. But anyways, that's what that is. R for ribose. And P for phosphate, if you're not going to remember what those things are. Okay, and then this is called the 5' cap. Now, they could have said like the front, but they don't call it the front end and the back end. They call it the 5' end and the 3' end. And then it'll be trimmed off and discarded. Okay, so the end, this is the really fun one. Okay, so what do they do to the end of it? Well, what's on the end of things that we're familiar with, like animals? What's on their end? They've got a tail, right? So this is cute. You have to put a tail on it. So it's going to have like a head and a tail. And the tail is called the poly-A tail. Poly means many, and A is adenine. So guess what the poly-A tail looks like? You just add this long tail. Now ours is not going to be long enough because these things can be like 200 A's long. And you go A. It's called the poly, and then A is kind of like in parentheses, A tail. Can be as many as 200 long. So that is how you put on the cap and the tail. Now, one last thing, bear with us, one last thing, so we've got to snip out these introns. And that's what we're going to do right here. And it's going to be simple and easy. What we want to draw is the thing coming in like this. And then the intron, the pink section, now these are going to look longer. This pink section is going to be a little bit longer than our drawing over here. We only had like 3 letters in it. We'll just pretend this is kind of a longer intron, because the introns usually are longer than just one codon. Okay, so they come in like this and they get pinched. There's, once again, a little machine, a little nano robot here that finds the section there, makes a loop. You know, like when you go to tie your shoes, you loop the thing around and you pinch it with your fingers. So this is the pink section that you want to get rid of. You can imagine it just brings them together, pinches, holds it, and then snip. You're going to snip that loop off. And the loop will go floating off looking like this. And they actually call it like a lasso or a lariat that will go floating off. And then this is our good stuff here. And then you just would glue these ends together. Once you snip that pink part off, you just stick the purples together. And we'll just draw a very generic enzyme worker robot thingy, and the name of this machine is called a spliceosome. That's a really nice name. I like that. When you splice something, you kind of cut it and stick it together again. Back in the olden days of cassette tapes, that was a major part of repairing those tapes. I can remember splicing. You would cut the ends of the little cassette tape, and then you'd very carefully try to tape them together again. So that's what this guy does. And this is back to not being a protein gadget, so it's called spliceosome, so it doesn't end with ASC. So you might go, maybe, I don't know. It's not telling me it's a protein gadget. In fact, it's not. It's an RNA gadget, just like a ribosome is made out of RNA. So the gadgets that are made out of RNA, they actually have a special name if you want to know it. You don't have to, but if you want to, it's called a ribozyme. Instead of an enzyme, it's called a ribozyme. That's kind of a nice word too, because you can think it's kind of like does an enzyme work? It's like a little worker, but it's made of the same thing that a ribosome is made out of. Okay, so that is the spliceosome right there. And that's it. After those things are taken out, the messenger RNA is ready to leave the nucleus, and you already know what will happen. It's going to go out into the cytoplasm, and it's going to meet a ribosome. The ribosome, it'll attach to the small subunit, and then it'll connect, and the amino acids will be brought by transfer RNA, and before you know it, you'll have a protein.